So, starting section 3. Section 3, we're actually going to cover 3 sensory systems. All right? So we're going to cover touch, olfaction, which is smell, and the gustatory system, which is taste. Okay? We're going to do bottom-up processing, and then we'll look at top-down influences for each of those. Okay? Sounds good. All right. And then our fourth and last section will be the auditory system, hearing. So we're jumping around a little bit in the textbook, but those chapters and things are on the Google site. All right. So your skin. We're going to talk about touch. We're going to talk about your skin. Your skin is the largest organ in your body. It's a good trivia question. Right? It's the trivia. Largest and heaviest organ in the body. So it's your skin as a whole is referred to as a singular sort of organ or structure. But what's the purpose of our skin? Well, we got to hold all this junk inside, right? So it maintains our physical integrity. It is going to provide protection from the environment. And most importantly, we're going to get information about our contact with the environment, the temperature of our environment, and we use our sense of touch to explore our environment in a lot of ways. And so we'll be talking about that. All right, so touch goes by a couple of names. So somatosensation is one, right? That means that's a fancy name for touch. Cutaneous senses, another synonym for touch. Cutaneous sensitivity is our sensitivity to skin stimulation. And as you guys are learning in your lab, we have thresholds for thermal sensitivity, right? We also have thresholds for mechanoreceptors and discrimination as well. We'll talk about that. So 3 main categories of the cutaneous senses. 3 main categories. We have mechanical touch, temperature, and pain. And we're going to focus on mechanical touch today, mainly, and we'll talk a little bit more about pain and temperature on Thursday morning. All right. So the somatosensory system is sort of the overall name for the skin, the receptors, the nerves, the pathways, the tracts, and then our main somatosensory cortex. All of that's enwrapped into the somatosensory system. So lots of different stimuli. You can have pressure on your skin, stretching of the skin, vibration, right? Texture, movement, and then we can have temperature, and we can even have mechanisms that detect when you damage your skin, and that's going to result in pain sensations. All right, so as I mentioned, we're going to focus in on cutaneous receptors, mechanoreceptors first. And so there are 2 layers of your skin. The epidermis, which is the top layer, and then a little bit deeper, the dermis layer. 95% of the skin on your body is what we call hairy skin. Even if you look at it and you're like, I don't think there's hair on that skin. There it is, very small, fine hairs. Right? The 5% that's not hairy skin is called glabrous skin, and that's found on the palm of your hands, the sole of your feet, and your lips and mouth. Everything else on your body is hairy skin. So the 5%, the glabrous skin, again, just found on your palms, your sole of your feet, and certain facial tissue. Okay. So that's just some definitions for you about things. So the mechanoreceptors are going to respond to mechanical touch, right, to physical touch of the skin. And main sort of categories of that are pressure, stretching, and vibration. And so we have 4 different mechanoreceptors that you need to learn. and they do 4 different things. All 4 of them together are contributing to our somatosensation. So the first one are called Merkel cells. They are disc-like in shape, and they're near the surface of the skin in that epidermis layer. The Meissner are look like a stack of pancakes or flat cells. And the shape of the receptor structures is very important, actually, to the type of stimulus that they're going to detect. They're also near the surface. So you can think about the 2 ones that start with M being closer to the surface, right? Associate the M with closer to the surface. The Ruffini and the Pacinian corpuscles are deeper in the dermis layer, right? So you can think about M comes before R and D in the alphabet, so it's closer to the skin. And then when we get deeper in, you have your Ruffini and your Pacinian corpuscles. The Ruffini are cylindrical in shape. They detect stretch. And the Pacinian corpuscle is encased in this onion-like structure. It's got these layers of cells surrounding it. All right, so there are 4 differences between the 4 mechanoreceptors. They do 4 different things. Okay? So the receptor structures are different, and that's going to allow them to respond to different types of stimuli, right? Disc, flat cells, cylindrical, onion-like shape. And because of those receptor structures, because of the shape of those receptor structures, and their location either near the surface or deeper in the skin, each one codes for a different type of stimulus. They have different neural codes. We'll talk about those. Because 2 of them, the Merkel and Meissner, are near the surface, they have smaller receptive fields. The ones that are deeper have larger receptive fields. So receptive field size is different between them. And combining their neural coding, their feature detection, and their receptor field size gives a unique perception for each of those 4. And we're combining all 4 perceptions. We don't perceive any one of these 4 independently, right? They're all coming together in our somatosensory cortex to provide an overall somatosensory perception. All right, so let's talk about transduction. In general, the innervating nerve that's cut, so this is the Pacinian corpuscle, right? Here's the nerve coming in there. I'm going to highlight it with yellow so you can see it better, right? And these somatosensory neurons, in general are pretty similar. What's different is how the receptor structure for each of those 4 mechanoreceptors interacts with that nerve. And so it really is the receptor structure that's giving it. So when there's movement, physical movement of this neuron, that's going to open sodium channels that allow that neuron to get depolarized and send action potentials. So movement, physical movement on the neuron is going to cause it to get depolarized, send signals. In the case of the Pacinian corpuscle, because of this receptor structure, it responds to the onset and then the offset of movement. So when it moves in the opposite direction, it also opens those channels and allows it to send action potentials. So there are 2 types of neural coding used by the mechanoreceptors. Slow adapting, or SA fibers, are going to respond continuously. They're going to be very slow to adapt. Why they're called slow adapt. They don't adapt. Right? So if I touch a Merkel or Ruffini ending and put pressure on it, they're going to fire action potentials continuously until I stop, and then they'll go back to their baseline. So the two slow adapting receptors are Merkel and Ruffini. What's different about Merkel and Ruffini besides their receptor structure? Yes. So we have one surface receptor, the Merkel, that's slow adapting, and one deep receptor, Ruffini, that is slow adapting. Then we have rapid adapting. Your textbook calls this fast adapting. It's the same thing. So you see RA or FA, it means the same thing. I'm just using some diagrams from another textbook that I like a little bit better. They call it rapid adapting. But rapid adapting fibers measure the onset and the offset of touch. And those are Meissner and Pacini and corpuscles. So when I touch, when I apply pressure to those two receptor types, they fire right at the start and then they adapt. They stop firing. And then when we release the pressure, they fire again. Can you see a usefulness for having two different types of neural coding? How would that be helpful? Yeah.
I'm thinking if it's like something you need to respond to quickly versus something that just happens every day, like if you touch a hot stove.
Okay. That's certainly true. That would be thermal in your example, not mechanical receptors.
It would be like wearing clothes versus like getting like pushed over, or I don't know.
Kind of, right? And we'll talk about the adaptation that happens with like wearing clothes. That's very much a top-down influence of the thalamus, right? But we have here, right, two different systems. Kind of, it's almost like when we talked about lateral inhibition in the eye. What was the point of lateral inhibition? To enhance edges, to enhance contrast. So here we have two receptors that are designed to fire when something starts touching us and when it quits touching us. It's kind of like enhancing the edges, right? And we also have two that are going to fire the entire time something is touching us, right? So it's like two different signals, one that's emphasizing the start and stop, and one that's the entire time that it's being stimulated. So that kind of makes sense to combine those, right? All right, so what defines the SA or the RA responses? And again, it's the receptor structure. It's the disc-like shape of the Merkel cells, or the stacked pancake shape of the Meissners, the onion-like shape of the Pacini corpuscle. That's what causes the rapid adapting and slow adapting. All right, so if we look at Pacini corpuscle here, and we were to isolate it and apply a constant stimulus to it, we would see that it depolarizes. So sodium comes in right at the start and then it stops. And while it's depolarized, it's sending those action potentials at the onset. And then when we release the pressure, the constant stimulus, there's another depolarization as it sort of rebounds and moves backwards, and we get another firing. If we take this and we strip off the onion-like receptor structure and we just have the neuron that was inside the Pacini corpuscle, and we apply the same stimulus, we don't get that adaptation. It depolarizes and continues firing. So it really is not anything to do necessarily with the neuron, it's the structure around the neuron that's allowing it to be slow adapting or rapid adapting. Does that make sense? All right, receptive fields. So slow adapting and rapid adapting fibers, we mentioned this on the other slide, that there's one of each for surface and one of each for the deeper receptors. And that results in a smaller receptive field, which we call SA1, and the deeper one is SA2, which has a larger receptive field. A larger receptive field. So these are more what we would say narrowly tuned, and more broadly tuned, so a larger area can stimulate those. Right? So Merkel cells are continuous response for a small receptive field. Ruffini are continuous response for a larger receptive field as they're deeper. SA1, SA2. The same thing for the rapid adapting. The Meissner are the small, narrow-tuned rapid adapting 1, RA1, and Pacinian corpuscles are RA2, or the larger ones that are deeper. So they respond to the larger receptor. All right. So it's this combination of, so it's receptor structure, providing either slow adapting or rapid adapting. Its location, which is either small or large receptive field. Right? And this combination of slow adapting or rapid adapting and receptive field provides unique perceptions. So fine details are detected through the slow adapting and small receptive fields. Low frequency vibrations are best detected by Meissner corpuscles. High frequency vibrations are best detected by Pacinian corpuscles. Stretch of the skin, best detected by Ruffini endings. All good? So these are the 4 things that are different. Receptor structure, neural coding, receptive field size, and that results in unique perceptions for each of those. All right. Now, when we get to the brain, we're combining all 4 of those mechanoreceptor perceptions into stimulating our somatosensory cortex. So here, we can take a monkey that we're going to record from neurons. We can actually isolate out those 4 different types of mechanoreceptors and record from each one. And this monkey's putting its finger on a drum that has 5 bumps on it. So every time that drum rotates across the monkey's finger, it's stimulating in this pattern, this plus-shaped pattern. And when we look at the action potentials that result from that, for this stimulus, the Merkel, the SA1, fine details, right? Their activity look very much representative of the thing that's stimulating them, right? Because they're firing constantly anytime the bump is there, and then they stop in between the bumps. So we get a pattern of activity that looks very much like the stimulus itself. Okay? With the rapid adapting, right, that's firing at the onset and offset, so it's much more messy sort of looking. And again, more fine details with the surface, smaller receptive fields, it looks a little bit more like this. With the deeper ones, because it's a larger receptive field, we can't see as much detail. It's like that whole time that something's firing. Right? And so it's putting all of these together in somatosensory cortex that gives us our overall perception of that stimulus. So in addition to the specific mechanoreceptor types having different receptive fields, right? Different parts of our body have different densities of those receptors, right? And that's going to result in different sensitivities or thresholds. So remember, high sensitivity means that you have a low threshold. You're very sensitive, you have a low threshold. Low sensitivity means you have a high threshold. And so it turns out that places that we have low thresholds, like our fingers, our hands, our thumb, our palm, right, our face, very low thresholds, have small and very numerous, dense receptors. Other parts of our body, like our arms and our trunk and our legs, have relatively low sensitivity, have relatively large receptive fields, and so the thresholds are higher. Why do you think we would want to have low thresholds for mechanoreceptors in those particular areas?
Yeah. We need to protect ourselves because if you're not thinking about the tip of your finger, you're going to touch, slip. If you're going to experience a stimulus that's more needed, you're going to want a pretty low threshold in case that stimulus takes place.
Okay, definitely, that's probably true for the face, right? Because we want to know with pretty accuracy where something's touching us on our face, right? For protection types of things. What do we use our hands for? Exploring our environment, right? If you want to feel something, do you go rub the back of your arm up against it? No, you reach out and touch it with your hand, right? So having high sensitivity and low thresholds gives us more information. Right? It's kind of like the phobia of our eye, right? We have more receptors, so we have better acuity. More receptors, low thresholds, high sensitivity. And then in places like our back and our trunk, we don't really care as much about locating where things are touching us, because it's not typically the way that we explore the environment. So, small, dense receptive fields, okay? Ability to discriminate very well on our fingers, tongue and lips, face, cheek, nose, and then other larger areas of our body like our back, our torso, our legs, our arms, much lower discrimination abilities. All right. So we have the skin covering our body, receptors all over it, neurons hooked up to those. We have to get the neurons to our brain, right? And so they have to come into the spinal cord. And what's surrounding the spinal cord? Bones, right? Your vertebrae. Right? And so these bundles of axons have to enter in between your vertebrae, to get into the spinal cord. And so there are 31 distinct entry points. So that results in 31 groups of neurons on the left and right of your body that are coming into the spinal cord. And we call the area, so if we have this group of neurons that's coming in here, right, to the thoracic area, where those neurons go out and innervate, we call that a dermatome. So a dermatome is a patch of skin that all the nerves from that are going into a particular spinal segment are sending information from. So it's highly organized, just like we talked about a retinotopic map of vision, right? Here we have a dermatopic map, a map of your dermatomes from your toes all the way up to your head. And we can map that out here, and so you can see that the T2 dermatome is going right across the top part of the chest and under the arm, right? And the lumbar 3 is your knee area. Right? And so these are organized from top to bottom, and they don't overlap really. They're distinct patches or regions of skin innervated by the nerves that are going into that particular part of the spinal cord. All right, so there are two distinct pathways. There's what we call the dorsal column or the medial lanistical pathway, and there is the anterior lateral spinothalamic pathway. A couple of differences between those. So the neurons are coming into the spine through the dorsal root. So that dorsal part is our back, right? The ventral would be towards the stomach. Right? And so in the dorsal columns, those are this is our spinal cord. We have our central canal there, the CSF in the middle of it. And starting at the bottom of our spinal cord, we're going to have your toes coming in. So, in general, the spinal cord is all axons, all myelin axons. It's like a big wire connecting signals into our brain, motor signals out of our brain. And it kind of looks like a butterfly shape where this area is gray matter, it's cell bodies and connections. These areas, these four areas, are white matter, pathways. So when your big toe neuron comes in, it's going to form a synapse, and then that neuron is going to go into the dorsal column and go up to the brain. And this would be from your right side, you have your left toe over here, coming in this side, and going up there, right? As we move up the spine, so now we get to your hands, what do you notice about how I've drawn this? It's bigger, right? Because we keep adding in axons. So your spinal cord's not the same size. It's wider at the top and it tapers all the way down to the bottom. So now we got our here's our toe over here near the midline. But now we have our arm. It's coming in on either side and adding. So we're building out from the midline in order of dermatome, right? These pathways. pathways going on. So the dorsal columns come in, travel up through, and these are called the dorsal columns, this part of the spine. Right? When they get to the medulla, up here, at the base of your brain, then they're going to cross over to the contralateral side, go to your thalamus, and then go to the contralateral somatosensory cortex. This is the mechanosensory pathway. The blue pathway is your mechanosensory. We have a separate pathway that comes in and crosses the midline where it comes in at the level of that dermatome and travels up through this pathway, which is our anterior lateral pathway. Crosses there and stays contralateral. This is our temperature and pain pathway. So we have a separate temperature and pain pathway from our mechanosensory pathways. All right, so medial lemniscal, again, comes in, travels up the dorsal columns, crosses over at the medulla, goes up to the thalamus, then to the somatosensory cortex. These are large myelinated fibers. They're fast conducting. We want to, as soon as something touches us, the whole job of the somatosensory system is to get information to the brain and let the brain decide what to do with it, right? So while we talked about a whole lot of processing in the retina, and that the visual signal coming into our brain has already been enhanced and changed in ways, not the case in the somatosensory system. We just want to get the signals up and into the brain. Right? Very little processing. The medial lemniscal system, this is our cutaneous, our mechanoreceptors, all four of those mechanoreceptors are traveling in this pathway. Crosses at the midbrain over to the contralateral. The spinal thalamic pathway, or the anterior lateral pathway, has a couple of names, same thing, right? These can have smaller unmyelinated fibers called A delta and C fibers. We'll talk more about them on Thursday when we talk about the pain system. They're slower conducting. They provide what we call non-localized cutaneous information, so not precise detail, but it's coming from a particular part of our body, region of our body, based on dermatome. And again, it crosses at the level of the spinal cord and travels up the contralateral side of the spinal cord. All right. So again, the lemniscal pathway, organized by dermatome, travels through the dorsal columns here, goes up to the, so the dorsal columns actually are like sort of further defined. Into sort of the lower half of your body and the upper half of your body. So S5 through T7, your toes up through about halfway up your midriff, right, are traveling in the most central part, which we call the gracilia fasciculus. And then the blue area above T7, T6 to C1 are traveling in this more peripheral or lateral area, and that has a different name, the cuneate fasciculus. So fasciculus means pathway, like the arcuate that connects the broken orifice area. So fasciculus is just a name that means pathway. And so the gracilla comes from the bottom part of your, bottom half of your body. The The cuneate comes from the top half.
Which side or which diagram thing?
Yeah, so this would be the gracilla, close to the midline. And then the cuneate would be further lateral, right? Because we're coming in and building outward as we're adding in more and more neurons as we go up the body. All right. Area of the thalamus. So far we've only talked about one area of the thalamus. What area is that? We talked about one sensory system. The LGN, right? Lateral genital nucleus. Now we have a separate area of the thalamus, the VP, ventroposterior thalamus, that's handling incoming information. So we're going to have a different nucleus of the thalamus for each sensory system. They're segregated in the thalamus into nuclei. Comes into the VP, forms a connection. What do you think's happening in the thalamus? What's its general job? It's not just a relay, it's a what? It's organizing. It's staying in the same dermatome level. Filtering, right? It's filtering. And so at some level, the thalamus it's like a gate deciding what signals get through and what signals don't get through. So right now, if I talk about your shoes on your feet, you can feel your shoes on your feet, right? A second ago, your shoes were still on your feet, but you weren't feeling them. That's the thalamus sort of blocking signals that it's deeming unimportant. But attention can be a top-down influence that lets those signals immediately through, right, when you think about a particular part of your body. But normally we're not paying attention to those, and so the thalamus is sort of dampening that signal and not letting it through. Okay. The spinothalamic system, these A delta and C fibers, come up through the spinothalamic pathway. They also go through the BPN, but they go to the cingulate cortex. In addition to, they also, some people often go to primary cortex. What is the cingulate cortex involved with? Does anyone remember? It's an attention area. Parts of the cingulate cortex are an attention area. It's also part of the limbic system. What does that mean? Emotion. So this is our thermal and pain pathway that's going up to stimulate our emotion. And so thermal and pain perceptions have an emotion associated with them. Right? When you touch something really cold, it doesn't feel good. When you have pain, it doesn't feel good. It evokes an emotion. So that area is going directly to the cingulate cortex as well. And again, we'll focus on the pain pathways and pain perceptions on Thursday. We're going to stick with sort of general somatic sensation today. All right, so we get up to primary somatosensory cortex, which is in the post-central gyrus, right? So you have the central sulcus that's separating the frontal lobe from the parietal lobe. So this is the very anterior gyrus of the parietal lobe is S1, somatosensory cortex. Right? And it stretches or reaches from the corpus callosum. So there's part of S1 that's on the inside of the longitudinal fissure, and then it wraps around and comes around into the lateral sulcus it's separating the parietal lobe from the temporal lobe inside here. Okay, where's your motor cortex? Come on, that's an easy bias question, Where's your motor cortex? It's in the precentral gyrus, right here, right? And so these things, often, right, we're using touch to either guide motor activity or to create actions, responses, motor responses to touch. So these things are right next to each other and they communicate heavily with one another. Okay, so I'm not really going to get into these different areas, but this primary somatosensory cortex is sort of organized into 3 different areas across each dermatome, and then we have a secondary somatosensory cortex that's sort of combining those incoming signals. That's really the level that I want you to understand. You don't really need to know the specific differences on that, right? It's important to know it's contralateral, right? So left somatosensory cortex is proceeding up to the right side of the body. But that information is shared between the 2 hemispheres. So if you run your finger across your chest, you get to the midline, you can't tell that now I'm going from my right brain to my left brain, right? It's all integrated through the corpus callosum. Again, organized by dermatome from your toes up to your legs, to your trunk, to your shoulder, to your hands, to your face, down here, right? From here, the somatosensory cortex is communicating with lots of different parts of areas of the brain. Right? It's getting combined with vision, as we talked about, with motor actions and touch. You'll notice that while this is in order of dermatome, we're dedicating uneven amounts of cortex to different parts of the body. And that creates what we call the homunculus, which is a map of how much cortex is devoted to different parts of the body. What parts of the body have the most amount of cortex devoted to? The face and the hands, right? Look, it starts from here and goes all the way down and around. All of that area, it's like, what would you say that is? 60% of the cortex is for your hands and for your face. Where do we have the most receptors? The hands and the face. So we're sending a lot more information to the brain, and we're devoting a lot more processing power in the brain to those particular areas. Very little to the rest of our body. Right? So this is organized by dermatome, spinal entry points. Right? But we have this magnification factor. And so the more cortex we devote to something, the better we are at processing that information. So again, we really want to know if something's touching our face, our head. We're also, somatosensory system is providing feedback about things, right? So when I'm talking is a very complex thing. We'll talk more about language a little bit when we get to the auditory system, but to move my lips and my tongue and open my jaw, I'm doing all these things in patterns to create sounds that are going to generate language that you can understand. And so I'm getting a lot of feedback to my motor system from the somatosensory system of how I'm moving my face, right? Same thing with our hands. We're exploring our environment. We have a lot of receptors. We're going to devote a lot more processing power to our hands. So, again, I made the point that the CNS area, the amount of CNS, is directly correlated with the number of peripheral receptors for those parts of the body. All right, a little bit about perception. So that's pretty much bottom-up signaling, right? We talked about types of physical stimuli. We talked about transduction. We talked about neural coding. We talked about the pathway from the receptor to the brain. And then we talked about primary cortex areas, bottom-up signaling.
Is there anything like the vision system where there's the subconscious pathway to the superior colliculus for touch, or not really?
Not really. Okay, so perceptions. We can define thresholds in lots of ways. We're talking about pressure, right? How much pressure do I need to put on my skin in order to detect touch? And it turns out that's going to vary depending on the body part, right? Depending on how many and dense the receptors are. But in our most sensitive areas, like our fingers, right, we can detect pressure of just 1 micrometer. Very, very small change in our skin. Right? Again, low thresholds, more sensitivity. It varies according to the body part that you're testing. Why does it vary? The receptor density, right? How many there are and how dense they are. Kind of hammering that home. Higher threshold for these areas, hands but not the fingers, like the back of the hand, right? So we have better perception, better thresholds for 2 reasons. More receptors and more cortex to process that information. You can think about the same thing that we talked about in the visual system. I'll try and show the similarities, but we had 6 layers of the LGN receiving part of the cellular pathway from our fovea, from our center of vision, and only 1/3 of that, 2 layers, from the peripheral vision, right? So things that are most important to us, we're devoting the most amount of resources to, both receptors and brain area. All right, so if we look at absolute threshold, right, we see that it's relatively the same between our left and right side across here. We have the lowest absolute threshold for our face, the highest for those other areas of the body, right? The other threshold that I showed you before was a discrimination threshold. Should have made that point better. Let me go back to that slide. So here, with this paper clip, it has 2 little points that are not very far apart, right? When we have small, numerous receptive fields, we have really good discrimination, right? Because they're touching different receptive fields, we can tell that there's 2 things and exactly where they are. Very good for our fingers, right? When we have large receptive fields, because they're overlapping and they're not, we can't tell necessarily if there's 2 points or 1 point. We have low discrimination. We can tell something's touching us, but we don't have a lot of information about it. That's why these are high, right? So this is discrimination thresholds and sensitivity to being able to detect details of touch. Yes, Doctor?
The colors over there, right? Is that you have high sensitivity as well?
Yeah. respond to those. I'll fix that for us. All right, the absolute threshold though, we go back to that other slide. We see the lowest absolute thresholds for our face, meaning where we can have the least amount of stimulation and still detect it. We have higher ones for our hands. So while they're good at details, they're not as sensitive to light touch, right? Okay. Where's the highest absolute threshold in our body? Which part of the body is the furthest from the skin? The soles of our feet, right? We spend a lot of our time standing on our feet. So if they were really sensitive, we'd be constantly getting tons of information coming in. So we don't really want them as sensitive because we're putting the whole weight of our body on our soles of our feet for large portions of the day, right? All right. Okay. Good. All right. Discrimination ability that I just talked about, the ability to tell if 1 or 2 points are touching our skin, is different. That was this thing, right? Is different than the absolute threshold. And so when we look here at 2-point discrimination, the face is still sensitive, is still the most sensitive, but now the hands, because of the high density of receptors, have very low thresholds. So we want to make sure that you understand the difference between absolute threshold and discrimination threshold. We have differences in thermal. Maybe you should be like, hey, introduction to my lab report, slide 28. All right? And so our thermal thresholds that you're measuring in the lab, right, also vary based on receptor location and density. And here we have a slight difference between warm and cool receptors. And again, we haven't talked about transduction and receptors and monomer signaling for that yet. We're going to do that on Thursday. But I wanted to go ahead and talk about the thermal thresholds. And we see where are your lowest thermal thresholds on this graph. For your face, right? Lips, cheek, forehead, right? And here we see slightly higher for our finger and even higher for our areas of the body, like our trunk and our legs and our arms. So, again, the head is most sensitive as a protective mechanism. And the feet are least sensitive, again, because probably for many, many years, right, our feet were very exposed to the environment. We weren't wearing shoes, we didn't have good shoes or clothing, right? And so over time, you would want them not to be as sensitive to thermal sensitivity. All right. There's also a difference in thermal sensitivity between glabrous skin and hairy skin. Glabrous skin, slightly higher threshold than hairy skin. So we can see that slightly higher here than it is here. And so higher threshold means lower sensitivity. And so you can think about that if we're reaching out, our glabrous skin on our palms of our hands and fingers, right? If that's our primary way of interacting with the world, we don't want it to be like necessarily as highly sensitive. Otherwise, every time we touch something, we might generate pain signals. Why is this the case? Well, the receptors that we'll talk about on Thursday are closer to the surface in hairy skin and are a little bit deeper in glabrous skin, which makes them not as sensitive. So here's an example of the location here. We see most of the glabrous ones are at a deeper depth of about 150 micrometers, whereas most of the hairy ones are in this 50 micrometer area. All right. Moving on. A little slower. Adaptation, right? Adaptation is where constant stimulation, our brain sort of deems that as not important information, and so we're turning down the neural signal. Turning down the neural signal. It's a decrease in our perception ability. Several things can affect that. The intensity. The more intense a sensation is, the harder it is to adapt to it. So if you have a loose bracelet on your wrist, it's easier to adapt to that than one that's very tight and constricting and intensely stimulating your wrist. It becomes harder to not pay attention to that. The area also matters. So when you're wearing clothes and they're stimulating large areas of your body, it's easier to adapt to them. It's easier to adapt to them than if you're wearing a ring on your finger that's tight. All right. A little bit about the organization of the somatosensory cortex. There's a high level of plasticity, meaning it's able to change. It's able to change. And what drives that change is experience, stimulation, use or no use. Use or no use. So again, in primates, we can come in and map the somatosensory cortex and see that parts of the S1 respond to the first digit, second digit, third, fourth. And even within the second digit, the tip of the finger is right here, right? And so if we give a monkey a discrimination task that they get rewarded for between different types of stimulation, of tactile stimulation on the tip of their second digit, and we give that monkey lots of training in that, the monkey gets better and able to discriminate better between stimuli over time. And the amount of cortex expands for that area with lots of use. We don't see the same level of plasticity in something like the visual system, but we see high levels of plasticity in here. Now, we have a limited amount of somatosensory cortex, so when we expand area 2, right, it has to take some of 1 and 3, the adjacent areas, in order to do that. So it's kind of like what we talked about with encoding emotionally important stimuli in the fusiform area. Normally, we're putting faces in there, but if we put greebles in there, we're using up some of the space for face. Right? Same sort of thing. All right. This is an example that changes plasticity due to use, right? Which is allowing us to do better discriminations, right? So if anyone's ever learned to play a stringed instrument, like the violin or the guitar, when you first start, it's very difficult. You're clumsy with moving your fingers. As you practice, your motor cortex is expanding and you're gaining better control over your fingers, more finite control. But your somatosensory cortex is also expanding to provide feedback and discrimination in those areas, right? Same sort of thing, and it's one of the things that helps us get better at doing that. Yes, Keisha?
Does this relate to why people with things in their sort of feel a sensation of their fingers and the plasticity?
Yeah, we're going to talk about that. Yeah. I'm getting there. So that's use driven. Now, no use or loss, right? Also, there's plasticity. So when, for example, somebody has an amputated arm and no longer has that big area of the cortex being stimulated by their fingers, right? What is the adjacent area next to that? You look at the homunculus, what was next to the hand? The face, right? The face on one side and the arm on the other side. And so as those adjacent areas of the brain come in and start to take over the cortex, so that cortex for the hand doesn't just rot or go away. The areas adjacent to it start to expand into it. And so now I have an area of the cortex that my adjacent arm is now stimulating that used to be the cortex associated with my fingers, and on the other side, parts of my face that are coming in to areas of the cortex that used to be my hands. And this is where the brain can sometimes get confused. It's like, okay, that area of my brain always represented stimulation from my hand, but I don't have a hand anymore. Why am I feeling that from touching my face or touching my arm? And when the brain gets confused, often it routes that signal to our pain areas. So just like when there's a large signal like bleaching your rods, routes it into pain, it's not really pain, it's just routing it into that area. This is the same sort of thing. And so there's several therapeutic strategies to help the brain recognize that, hey, that part of my brain that used to get input from my hands now gets input from my arm. And that's really my arm and not this phantom feeling coming from my hands that's not there. Does that make sense? Okay. So when we're looking at these cortical receptive fields, right, we have two different layers. So we have the number of receptors in the periphery, whether they're small and dense and non-overlapping or large and overlapping, coming in by dermatome. And then we have it based on receptive field. And these receptive fields use the same principles. We still have to have convergence. We have way more somatosensory receptors on our skin that we can funnel neurons into our spinal cord and up our spinal cord. Now our spinal cord is way bigger than the million neurons in the optic nerve, right? But it's still limited in its resources. So we have the same principles of needing to converge the signal to get it in the brain. And once we get it in the brain, we can diverge it back out to lots of neurons in the brain, and we diverge that more so for our hands and our face to give it more processing power. So somatosensory receptors utilize that center surround neural coding in order to get two signals again for the price of one neuron. So because we have baseline firing rate, right? We saw that in the visual system. I used to teach this first. I need to change that. But we see the same sort of thing from the visual system we see here, right? So the center can be inhibitory or excitatory. It's just like the visual system. It doesn't really matter. The point is that you're able to discriminate between something touching here and something touching here with one neuron. With one neuron. And this is going to allow for better discriminations with lower numbers of neurons. All right. Just like the visual system, we have columns in the somatosensory system within the dermatomes. And so if this is my digit 2 and my digit 3, there's a column, and as I go deeper into the somatosensory system, all of the neurons in that column are associated with D2 stimulation. So it's the same sort of column organization, except now we're organizing it by body part across the homunculus. Within that, we have, this is where we're combining those 4 mechanoreceptor sensations. This is where they're getting combined in that column. So we actually, within that digit, have areas that are receiving slow adapting and areas that are receiving rapid adapting next to each other. And they are getting combined into a singular perception, at that cortical area. So again, columns are coding very specific features, just like in the visual system, columns were coding for different angles of lines. Here we're coding for different types of vibrations and details of touch. But specific features. We're breaking down whatever's touching me into 4 distinct features, sending those signals to slow adapting and rapid adapting neurons up to columns in that dermatome. All right, so this is where all that's getting combined. And so again, if we look at rolling these letters and we look at details with slow adapting, right? And then the rapid adapting, we get more of the contrast, right? When we layer those on top of each other in a combined area, right, we're getting the full picture of what that perception is, right? And so we have those feature columns in S1, and those feature columns are getting combined into that overall perception in S2. So that's the main sort of difference between primary and secondary somatosensory cortex, is that primary is still organized by slow and rapid adapting columns, features, and those columns are coming together in secondary S2 to generate our overall perception.
Can you say that one more time?
Yeah.
Any difference between primary and secondary?
Yeah, so in primary, we still have columns for slow adapting and rapid adapting, and they're doing separate feature detection. And then those features are getting combined in S2, secondary, as our perception. So our conscious somatosensory perception is coming out of S2, but it's the result of S1 feature detection. Just like in the visual system, we don't really perceive V1. It's those higher secondary areas that are combining the features to give us our overall visual perception. Yes?
What is the 3B RN PSA?
Yeah, you don't need to know those numbers. It's more technical stuff. If you will just understand it at a level of primary is still organized in columns that are detecting features that are slow and rapid adapting, and secondary, adjacent to that, is combining those. This is actually like the depth of these like 3 A and B, like deeper in the cortex. It's more information than you guys need to worry about. All right. We have columns. So again, so we have receptive fields here, just like we saw in the eye, right? There we go. Bunch of receptive fields, very dense and numerous on our hand, right? Sending signals up to our brain, and then we devote a lot of brain area, that magnification factor, a lot of brain area is devoted to processing that. And so we can diverge the signal and have then columns that respond to different line orientations. So that we have a column in the brain that is receiving input from all of these receptive fields, but not from the yellow ones. And so as we rotate a line across there and activate more, right? So when we're here, not as much action potentials because we only are. So in a lot of ways, the brain's taking similar organizational features and using them across different sensory systems. So we actually get a tuning curve for something that's horizontal, and then we have another column that's looking at diagonal lines, and another column looking at. So instead of lines, we're talking about things touching us in different orientations. We also have movement and texture-specific ones. So we have all of these different types of features that are represented in columns in that one dermatome, or for that one particular area of the body. So we can find neurons that respond when we move from right to left across the finger, direction specific, and they don't respond when we move from left to right. And there's even more than this. But the point of this is that the organization of S1 is in columns that detect specific features. Those features can be very simple in the form of SA and RA stimulation, but can also diverge and create features like movement or direction-specific edges. All right, last little bit. We've talked about attention a little bit with the thalamus, right? So here we're giving a monkey 2 taps. It's putting its finger on that drum, and when it's detecting particular stimuli, it can press the lever and get rewarded. Right? That's tactile attention. But then we can also flash things on the screen, and now when we flash things on the screen, the monkey is getting rewarded for responding to visual stimuli and not getting rewarded anymore to the tactile. But its finger is still on the drum, and so the bottom-up signaling is not changing at all in this experiment. Bottom-up signaling is not changing at all, but we're redirecting the monkey's attention to either vision or tactile. And if we look at that somatosensory area associated with the monkey's finger, when it's having to do the tactile response, there's a lot of brain activity. But when we switch over to the visual reward, the brain activity goes down, even though we're still stimulating it the same way bottom up. So this is the difference in the thalamus redirecting attention away from paying attention to what's happening with my finger and paying attention to what's happening with my visual system. So attention and other top-down influences, right, are changing what activity is getting through the VP of the thalamus to our perceptual areas of the cortex. You good? All right. So in addition to detecting touch, mechanical reception, I also talked about a role of feedback for the sensory system. And so our sense of kinesthesis is our awareness of our body in space, of our body position and movement. Where is my hand? Where is my arm? Right? And it's a feedback system primarily used by the motor system for helping us control our muscles and helping us maintain our posture and maintain our balance. And so there are particular receptors called proprioceptors that are providing this kinesthesis, right? And the 2 proprioceptors that we're going to learn about are muscle spindles and Golgi tendon organs. Separate from the mechanoreceptors. This is a whole separate part of the somatosensory system. So kinesthesis is perception of our body position movement. Haptic is perception is using both our mechanical, our cutaneous sensitivity, our mechanical receptors, and our kinesthesis in order to identify objects. So haptic perception is the ability to identify objects combining these systems. All right. Proprioceptors are providing feedback about the stretch and tension of our muscles. This is kinesthetic system. And so we'll start with the muscle spindles. So put the S of spindles with stretch. Spindles detect stretch. When is our muscle stretched? When it's relaxed, right? Not when it's contracted. We stretch out the bicep when we extend our arm, when we contract the bicep, right? When we pull our arm and when we initiate that. So spindles are sending a signal that say, hey, you're not using that muscle. It's stretched. And so it's allowing, it's saying basically if you want to initiate a contraction, go ahead because you're not doing that right now. The Golgi tendon organ detects tension. Put the T of tendon and tension together. And we have tension in the muscle as we start to contract it. And so now the signal is, hey, you're using that muscle. Anytime you want to stop doing that, you're welcome to stop doing that. It's a signal that allows us to know we can stop and hit, stop contracting. These two together help us know, is my arm out, stretched out, or is it contracted and curled up? Helps me know where my arm is in space. So we have a signal, I'm not using my muscle, comes in, and then we get a motor signal out to contract it, and when it gets contracted, that signal goes away, and now we have a signal from our tendon organ, saying, hey, you're contracting that. Anytime you want to stop sending those action potentials, you can stop doing that. All right, so I'm gonna lead with this. I'll lead with this slide, these two slides. The alpha motor neuron is our contracting neuron for the motor system. The 1A sensory neuron reports muscle spindles, so think about stretch coming before tension. So A comes before 1B, which is going to detect tension. So we have two different types of sensory, kinesthetic sensory neurons. The 1A are hooked up to the spindles, the 1B are hooked up to the Golgi tendon organs. As we start to contract our muscle, so I fully stretched, now it's not as stretched. That causes the spindle to get like slack. So there's actually this thing called a gamma motor neuron that kind of tightens up the spindle so it can still, I need to know I'm not fully contracted, I'm just a little bit, right? So that gamma motor neuron essentially like keeps the spindle shaped well enough that it can continue sending those signals. All right, I'll pick up here on Thursday. If you can come to class a few minutes early, I'm going to have a thermal demonstration that you can do. Put your hands in a hot and cold bucket of water. It's kind of cool. But I'm not going to use a whole lot of class time to do that. So if you want to do that, try to get here a few minutes early. It'll be a demonstration that we talk about in the class.